All right, thank you, Anne. So just a quick history of the SeaTech program. It was started back, I think, in like 2006 with an NSF grant that was with the Scripps Acoustics Lab and the Ocean Institute. And we came online at Mount Edgecombe in about 2009 with Josh Jones and Ethan Roth and two students who were willing to take the leap and try something new. So we have been doing this now for 11 years. We've had over 50 students in the program. And one of the main purposes that I feel that this class meets is often in classes, we do a lot of lecture lab. We fill students up with knowledge. And I often bring experts into my classroom, and these experts say, if you go to school for a long time and then you do all these things, then you could be somebody like me. And I think we're just really fortunate to be able to work with Dr. Hildebrand in the Whale Acoustics Lab, because his idea of science is that we should do it now. And one of the main purposes and the goals of the SeaTech program is to get students to do authentic research and to be able to present their findings that are new to the world via these types of seminars or a presentation or at some sort of a conference, and share our world of scientific research. Also, a side benefit of this for the students is that they create a great network for themselves of scientific members, people in the scientific community that they can know and that might help them someday get a job in this. And then the last benefit and goal of the program is to really foster this love of the ocean and the animals that live in and around it. And I think that all of the students, as they come into this, have some sort of a passion for marine biology or ocean science, and that's why they venture in here. So it's great to work with a group of students who choose to be in the room, and they work really hard. And we could not do this without some of the partnerships that we have. We've had, Ann Simonis and Natalie Postalgian that have been with us the entire way. They meet with our students at least once a week, usually more like twice or three times a week, and help us to analyze this fantastic data that's provided by the Whale Acoustics Lab. So this year we have had 11 students with us. I think we have 10 of them online. Just some disclaimers before we get started. I will be trying to run the presentation from my location, but just know that we might have some small lags or technical difficulties as the students are in 10 or 11 different other locations. Mount Edgecombe is a state-funded public boarding school, and so the students, when they are here, live in the dorms. And living in the dorms, as you've all been through college, you know that it can be a challenge at times, living with a large group of other people. So it's a real status symbol for our students to say how long they've been here. So if you hear somebody say, "I'm a 4-year senior," that doesn't mean that this is their 4th time trying to be a senior. It just means that they've survived the dorms at Mount Edgecombe for 4 years, and they're really proud of that fact. So let's see here. We will have some sounds that we play during the presentation, and we've done some testing, but we found that some of the sounds might be very faint for you. So just keep that in mind as we're going through and playing some sounds. You may not be able to hear them very well. So without further ado, I'm going to start sharing my screen, and we will start this presentation. All right. Oh, okay. Is everybody seeing the screen share?
Yep, I can see it.
All right, fantastic. All righty, so Kevin, we're ready for you.
I'm muted, but I am on video, just so you know. But you're not.
My name is Kevin Monagold. I come from Kotzebue, Alaska, and I'm a 3-year junior at Mount Edgecombe High School.
Hi, my name is Amaya Edwards Davis. I am a second-year senior from Mount Edgecombe High School. I grew up in Barrow, Alaska, and that's the city beneath the whalebone arc. Yeah. Something about me is that I love being a part of different programs and trying new things. So I was also a part of a 3-month college program called Raw High here in Fairbanks, and I did a 4-year geology summer program that allowed me to travel to different parts of Alaska and the lower 48s. For the future, I plan to pursue a career in animal science, which I put a picture of me feeding a giraffe at the San Antonio Zoo. And I chose to do SeaTech because it was a class for me to learn about marine mammals and gain insight on animals beneath the water. So, yeah.
Jaden, are you on?
Yeah, hello, my name is Jaden Frost, and I'm from Togiak, Alaska, and I'm a senior at Mount Edgecombe High School. The reason why I joined SeaTech was because my friends talked to me about the class and it sounded interesting. Also, this class gave a perfect opportunity to learn more about marine mammals. Since Togiak is a village near an open ocean, we are constantly surrounded by marine mammals. My future plans is to go to the University of Alaska Fairbanks and go to the medical field to become a paramedic.
My name is Jesse Hansen. I'm a 4-year senior from Sitka, Alaska. I spent the first 16 years of my life growing up on a sailboat. I've always been around the ocean, been on the ocean. It's always been a very integral part of my life. And so I took SeaTech because I had that foundation, and I feel like the knowledge of marine mammals and their environment, how they operated, was missing from my life and from my experience.
I'm Zane Mahoney. I'm a junior from Sitka, Alaska, and I've always been interested in marine biology. I've always thought it was just so interesting because we know so little about the ocean.
And Zane is my son.
My name is Haley Osborne. I am a senior from Nome, Alaska. I work as a commercial fisherman in the summer and hope to pursue a career in welding.
This is Leah Lakanoff. She is from Anchorage, originally the Pribilof Islands, and she's unable to be with us today.
Can you guys hear me good?
Yeah.
Because of my microphone. Okay. Yakwe, what I just said was a Marshallese greeting that means happiness to everyone. My name is Tiana Capelli, and I am a second-year sophomore at Mount Edgecombe High School. I was born and raised in Fairbanks, Alaska, and I enjoy going out on our beautiful land and water to collect our natural foods. I also enjoy traveling the world and attending youth missionary camps.
Hi, my name is Sophia Kamoli. I am a senior at Mount Edgecombe High School. I live in a small town called Hoona, located in southeast Alaska. Some of my favorite things to do are go fishing with my family and go out on boat rides. My priority after high school is to become an EMT and a firefighter in my community. One reason why I chose to take SeaTech is because my older sister told me about it when she was a senior, and it sounded very interesting to me because I love science and I've grown up by the ocean, so I thought it'd be interesting to learn more about whales and their lifestyle. One fact about me is that in the summer, I'm a whale watching tour guide for humpback whales and killer whales, which is the picture on the bottom right.
My name is Noah Andrew. I'm a junior from Bethel. I enjoy wrestling, boat riding, and anything STEM related, which is why I was interested in this class. After high school, I plan to attend Embry-Riddle in Prescott, Arizona, where I plan to be a computer engineer.
I'm Reese Banky. I'm a junior from Nome, Alaska. I've lived there my entire life. Nome is cold, dry, and windy in the winter, and warm, dry, and windy in the summer. Some of the things I enjoy doing are snowboarding and cross-country running. I was convinced to take SeaTech by my older brother. He thought it would be a class I'd very much enjoy, and he was 100% correct.
Can you guys see this? See his screen?
Yeah.
Okay, I can't see it. Okay. Today we will be talking about the passive acoustic monitoring for odontocetes in the Gulf of Alaska from April to September 2019. I want to include that there are two sites that we will be talking about today. Site CB, which is located at the continental slope, and KOA, which is further offshore. It's hard to visually study marine mammals in their habitat. There are multiple factors that come into play when trying to visually study marine mammals. Today we have the technology to hear the mammals and other species underwater without any human interaction. Although we are focusing only on odontocetes, there are numerous other species of marine life. With the changing conditions of the marine mammals' habitat, the Gulf of Alaska has effects from climate change. The effects of climate change that we can see is the warm blob, warming temperatures, rain shifts for some marine mammals, and less productive waters could mean less marine mammals. From the animals adapting to the changing climate to humans hunting entire animal species to near extinction, these marine mammals are at risk. Monitoring these species closely with acoustics will help us better understand what is happening and future planning on important decisions to be made relating to marine mammals. Odontocetes have teeth. They also have the ability to produce sounds underwater. The picture above is a sperm whale head. These animals have what we call melons, which you can see as the yellow blob in their skull, where it is located on the top of their head. The melon has the ability to produce and radiate sound in the form of a click. The clicks they produce bounce off things in the water around them. The odontocete receives sound back from the environment through their jaw, which is connected to the inner ear. On the jaw, a fatty substance helps sound travel more efficiently to the inner ear. Odontocetes use echolocation for navigating through the water and hunting prey. We have a couple of goals in this study, and here's what they are. We want to know when animals are at the site. The presence of marine mammals is crucial to understanding them. We also want to know when animals are present during anthropogenic sound, humans are near. Marine mammals behave variously to different species, but we want to focus on what happens when killer whales are nearby. Killer whales are known to be the apex predator of the seas, so we want to know what other animals behave like when they are around, as well as not around. Finally, we want to compare previous years of data to this year's data. Comparing data is important and may yield crucial results.
Throughout this section, I'll be sharing with you some of the methods we used to collect our data information. First, we'll be going over the location of where the data was collected. There were two sites in the Gulf of Alaska, CB and KOA. CB is right on the continental slope. At this site, it contains a lot of marine mammals or marine mammal occurrences. KOA is located southeast from CB. These two sites are also placed by Kodiak Island. Kodiak Island, Alaska. Kodiak is a popular place to go fishing. The instrument that was placed at CB was 970 meters deep, and the instrument that was placed at site KOA was 1,000 meters deep. The instrument that we used was called a high-frequency acoustic recording package, or a HARP. The HARPs had a sampling rate of 200 kilohertz. The HARPs are 10 meters above the seawater, and the flotation at the top are like buoys. They help the researchers find where the HARPs are once they come back to collect the data. The hydrophone are just like the ears, they collect sounds. These sounds can be whale clicks, explosions, anthropogenic noises, and ambient sounds. As the hydrophone is collecting the sounds, it transfers the sounds to the data logger, the brain. As the data logger collects the data, it stores the sound in the RAM. Each HARP monitored about 156 days between April and September 2019. Triton was a piece of software that was developed by Sean Wiggins that allows us to see sound. Triton was also our main source that we used to identify detections on. On the right, this picture shows the window of LTSA on top, spectrogram, time series, spectrum of Risso's dolphin clicks. We'll talk a little more about those later. We also used the logger in Triton to mark the start and end time of each detection.
All right, this is a spectrogram, a tool used to visualize sound. That has a few clicks on it in the red circle. Spectrograms have time on the x-axis and intensity of sound shown in color on the y-axis. Using this specific information, we can find out about these clicks so we can identify species. We know based on the literature that these identification methods are reliable. Based off the species ICI, inner click interval, peak frequencies, and click size, I can ID the species. We'll explain the tools used for identification in greater detail later. This is a flowchart made by Mr. Mahoney, my dad, using several different pieces of literature. We use this to narrow down what species we could be looking at. This shows peak frequency as well as range in the spectrogram, as well as the plot on the right, a spectrum. As you can see, this click starts roughly at 40 kilohertz and ends around 90. It's most intense through those frequencies. The spectrum shows this information as well, where frequency is shown on the bottom axis and intensity of sound is shown on the vertical axis. This is a time series showing 1 second of clicks. On the x-axis is time and amplitude is on the y. Because it shows 1 second, we can determine inner click intervals. Notice that clicks are about every 1/10 of a second, meaning that they are 100 millisecond clicks. We can use this identification chart with this information to match this species ICI. The time series shown on the top is zoomed in on 1 click. It shows a click with many cycles and a long duration. Different odontoces produce different types of clicks. On the left is a spectrogram showing a click with upsweep. This is typical of beaked whales. I've added a line to highlight that upsweep. The spectrogram on the right has no upseat, which is typical of dolphins. Notice that each of these clicks have different durations, with beaked whale clicks having longer durations than dolphin clicks. These are click characteristics we use to narrow down what species we could be looking at.
So, anthropogenic noise is any sound that is generated by a non-biologic source, anything like ships, sonar, things of that nature. And so why do we care about things that aren't whales and dolphins that are existing in the data and the ocean? The main objective of our data gathering is to understand animals and their behavior. And behavior is dictated partially by environment, thereby anthropogenics, a big part of the ocean environment, especially at a site like CB and KOA in a heavily trafficked region of the ocean. Now, in some cases, this is more severe than others. In the case of MFA, or mid-frequency active sonar, primarily for a military application, has been linked in some cases to injury or death in some species due to its abrasive nature. The anthropogenic sound may interfere with important natural behaviors such as foraging, resting, and reproduction. And some types of anthropogenic sound, i.e. military sonar, as I previously mentioned, have been linked to direct injury or death in some species. Further, anthropogenic sound indicates that humans are present, so looking at the relationship of anthropogenic noise with the sounds of species of interest may tell us how behavior of the animals is influenced by human activity. Now, the two main types of anthropogenic noise that we found in our data were ships and MFA sonar. Now, the first of which, ships, there were three main types that were that could be heard at the hydrophones. However, they were all logged under the same category: fishing, transportation, and commercial ships such as shipping and tanking and things like that. Now, if we listen to one of these sounds of a ship, you can tell that it's fairly hard to differentiate what type of ship, what type of engine that ship is carrying. So they were all logged as just an occurrence of a ship. And so you can see here that the characteristics of a ship are a swell in cavitation noise and low frequency noise and an echo sounder band. So all ship types appear the same on the LTSA. However, we were able to use a program, an automatic logger that detects ships without detecting them manually by a human because of their specific nature. There isn't a lot of other things in the ocean that sound exactly like a ship, and you're able to isolate that repetitive droning engine noise and use a program to automatically detect that kind of thing. However, echo sounders, the light banding you see around 30 kilohertz, we did manual detections on. Now, the other type of anthropogenic noise is MFA sonar, which has a strictly military application. So MFA sonar differs from things like echo sounders and other types of passive sonar in that active sonar, by its very nature, sends out a very loud, abrasive sound pulse that bounces off anything in the water and comes back to the sonar receiver, painting you a picture of whatever it is in the water. For military application, namely submarines. So if we listen to a recording of some MFA sonar on the data, it's very easy to understand how that could be abrasive and disruptive to really anything going on in the ocean. Now, here we have a 24-hour plot of anthropogenic noise at site KOA. So as this is the first 24-hour plot I'll be going over what it's showing. So on the bottom on the x-axis, we have time in 24 hours, 10 hours behind UTC time to give us Alaska time. And on the y-axis, we have date from the drop-off to the collection of the hydrophone. And so a band of sound displayed on this graph is wherever it lands on the y-axis is the date that it happened, and however long it is how long it continued for. So in lighter orange, we have echo sounders, and in darker red, we have ship noises. And you can see in the first part of the deployment of the hydrophone, there's almost strictly echo sounders. And moving forward, there is a more of a prevalence of ships, perhaps indicating some sort of seasonal movement between types of ships trafficking the region. Moving on, we have a weekly presence graph showing the presence on a weekly basis this time of ship detections at Site KOA. So this is the very same data pictured in the graph previously. However, it is on a weekly basis instead of showing time. So here really you can get a better picture of overall what is going on, overall how much traffic is there in the region. However, it's a lot less specific to when these things are happening, what times they were going on. It's simply shown as a bar graph. And so on the bottom, on the x-axis, we have dates this time, and on the y-axis, we had cumulative hours per week that there were detections at the site. The other two things on this graph that are different from the previous one are the effort dots. So the hydrophone was not dropped off Sunday at midnight, the beginning of the data, and was picked up Saturday at midnight, the end of the data. It was deployed and picked up whenever they could get out there. So the effort dots are the actual start of the data. So it's showing that there may be some discrepancy in the first week and the last week of data, as it is not complete. So moving on, we have another 24-hour plot, this time showing ship detections at Site CB. Now, as you can see, the Gulf of Alaska was a very, very popular place to be right around the middle of June, and we can see this very clearly in a 24-hour plot. As I said before, strictly ship noises are highlighted in red, echo sounders in orange, and we only had one occurrence of mid-frequency active sonar in the CB data, and that is pictured in blue for around 4 hours. Now moving on, we have the same data pictured in another weekly plot. Now, as you can see, the same strong band of activity is shown this time in a bar graph, and the effort dots are all present. However, it's not as much detail as a 24-hour plot can show, simply because you're compressing the time onto a weekly basis instead of an hourly basis.
Reese, you on?
Orcas, or commonly known as the killer whale, is the largest member of the Delphinidae family and is a top marine predator. Killer whales are easy to identify visually by their black and white bodies. You can see the species distribution on the light blue sections of the map shown on the slide. The map on the lower left shows that killer whales can be found in every ocean in the world. They are the most widely distributed cetacean. The killer whale has three ecotypes: transient, resident, and offshore. Resident killer whales live in coastal waters with more restricted ranges than the other ecotypes and feed mostly on different kinds of fish. Transient killer whales live in the coastal waters throughout their range and have a much more varied diet, eating other marine mammals such as seals, sea lions, dolphins, and other whales. The final group, offshore killer whales, reside in deep offshore waters and eat fish and sometimes sharks.
Killer whales have a wide range of acoustic behaviors. Echolocation clicks, buzzes, whistles, and pulse calls. Echolocations are used for echolocation. Buzzes are used for close-up echolocation, possibly for hunting or foraging, as buzzes are clicks with incredibly small ICI, while whistles are used for communication and social interaction. The clicks of these whales are higher in frequency compared to sperm whales. The frequency range of killer whales don't reach as low as sperm whales. We can use this to distinguish the two species. The difference between killer whales and Barrett's-beaked whales are hard to tell, as both species make sounds around the same frequencies. Also, Barrett's-beaked whales can make whistle and buzz sounds similar to killer whales. To differentiate these two species, we would look at an individual click. Killer whales' clicks don't have upsweeps, and their clicks are short in duration compared to Barrett's-beaked whales, with little up and down cycles shown on the time series on the lower right figure. Killer whales make cool sounds that makes detections interesting, considering most of the detections are clicks. Shown on the spectrogram here are examples of pulse calls and buzzes. If you hear this and you're in the water, it's too late.
Something right over the spot where I'm supposed to play the sound. Stand by.
You can move it out of the way, I think.
Yeah, I'm trying to. Let's just do this.
Pulse calls are used for maintaining familial bonds and communication, while buzzes, as I've mentioned, are for close-up echolocation. This combination of sounds are typical of killer whales, making it easier to distinguish from Barrett's-beaked whales, as Barrett's-beaked whales do not make pulse calls.
Here's a 24-hour plot with time on the x-axis and date on the y-axis. This is from the KOA data site. The killer whale detections are shown in blue. You can see on the data that almost all of the killer whale detections are from June to late August, aside from a few outliers occurring in April and September. Here's a figure showing the weekly presence of killer whales at the KOA site. Like the previous data shown, you can see that there's a stronger presence of killer whales from June to late August. The peak amount of killer whale detections was in early June, reaching a total time of almost 6 hours per week.
Here's another 24-hour plot. This one shows detections of killer whales at Site CB. Killer whales were detected right away when deployments at this site started and were common through the end of July. Again, the weekly presence at this site started off strong in through July, with peak detections in late May, and little activity from August till the end of the deployment. Notice that there were up to 60 hours of activity in a week around June. This is 10 times more than the peak weekly presence observed at KOA.
Here we have killer whales and anthropogenic noise in Site KOA. Ship detections are shown in red, echo sounders are shown in orange, and killer whales are shown in blue. Here we do not have any moments where they are heard at the same time. This suggests that both killer whales and ships have no correlation to each other at Site KOA. This is similar to the plot we've shown before, but here we've added MFAs in teal. Here in Site CB, we have a few moments where killer whales and anthropogenic noises can be heard at the same time. You can see a few at 6 p.m. on the 18th of June, as well as 1 p.m. between 18th of June and 2nd of July. We observed times when both killer whales and ships were not present throughout August. We're not sure why this could be, but it is interesting to point out that the end of summer and fall of 2019 was very warm in the Gulf of Alaska, which could affect animal behavior.
Sperm whales, also called Physeter macrocephalus, are the largest toothed whales of the odontocets, weighing up to 40 to 50 tons. Sperm whales are found at great depths of a few thousand feet, holding their breath for about 90 minutes. They are located all over the world, pictured on the map at the bottom right in the dark blue. They feed primarily on large squid and fish, also pictured lower left, which makes them hard to find, especially with their gray skin that blends in with the water. When identifying sperm whales in our data, it is important to remember that their clicks are very distinctive. When listening to sperm whales, their clicks are always consistent with only seconds apart from one another, almost like a metronome. They can also make slow clicks. Scientists suggest that slow clicks happen when they are surfacing, but not when they dive. Codas can be identified as social clicks when they interact with one another. Slow clicks appeared quite a few times in our data, but we did not find any codas. This is an LTSA and spectrogram of sperm whale clicks. The LTSA at the top shows low frequency sperm whale clicks at around 10 kilohertz and below, which is the red-orange area on the bottom.
Go ahead, Sophia.
Okay. The spectrogram at the bottom shows those clicks zoomed in so that we can see each individual click. Their peak frequency is usually around 10 kilohertz, and they can go lower, but no more than 20 kilohertz. The spectrogram also shows their distinct interclick interval of about 1 click per second. This figure shows the occurrence of sperm whales at site KOA. Each blue mark you see is sperm whale activity. The x-axis shows the hours of day, the y-axis shows the date of time from the first day of research in 2019 at the bottom to the last day at the top. As you can see, there was a decline in sperm whale activity from early May to mid-June, and also near the top from the end of August to the end of September. As you can tell, this figure is a little different. While they both show sperm whale activity, this graph shows the occurrence of sperm whales in the data from each week they were identified. One interesting thing to note is that there was 1 week in August when there's nearly 160 accumulative hours of sperm whale activity, which is about 95% of the week. At site CB, pictured above, sperm whales were present nearly the entire time, with higher presence compared to KOA. Lastly, it is clear that there were substantially more sperm whales detected clicking at site CB compared to site KOA. There were several more weeks at site CB compared to KOA that had nearly 160 hours of sperm whale activity.
This is the weekly presence of anthropogenic sounds in sperm whales at site KOA. As you can see, there are hotspots in both the sperm whale and anthropogenic sounds. These results show some interesting trends. Something that we've noticed is that when there's a peak in anthropogenic noise in May and August, there's also a peak in sperm whale detections. We are not entirely sure why this correlation happens, but there is evidence that sperm whales might be attracted to anthropogenic sounds, as they have been observed feeding off gear from fishing vessels. This is the weekly presence of anthropogenic sounds in sperm whales at our other site, CB. You'll notice in mid-June that during the peak of anthropogenic noise, there are no sperm whale detections. Sophia is going to explain why we think this happens.
Masking is a term to describe low or high frequency sounds being overpowered by ambient or boat noises or even other whales. This is an LTSA showing ship noise over 130 hours. If there were sperm whales, we would have expected them to have a frequency of about 10 kilohertz and lower. But if you look at the frequency band, you can see anthropogenic noise, making it very hard to identify the lower frequency sounds like sperm whales. In the plot at the right-hand corner, you can see weekly presence of sperm whales in the gray and ships in red. There were 3 weeks in June where there was a lot of ship noise that was probably masking the sperm whales at where they were.
Steineger's beaked whales are distributed across the North Pacific Ocean, along the Aleutian chain in Alaska, in California, Southwest Bering Sea, and the Sea of Japan. Our 2 sites were located in the Gulf of Alaska. Steineger's beaked whale have distinguishable clicks. Their clicks have an upsweep of a peak frequency at 45 to 73 kilohertz and have an average time of 90 milliseconds between each click. LTSA, long-term spectral average. The x-axis is time in hours, and the y-axis is frequency in kilohertz. And the second y-axis is color code for the intensity of the click. The more blue is the cooler the click, and as it gets more red, it shows that the click is more intense. Steineger's click, as shown, starts a little above 40 kilohertz and goes all the way up to about 95 kilohertz. Click characteristics. Spectrogram. The x-axis is time in seconds and the y-axis is frequency. It is the exact same as LTSA but shows a much shorter time span. Time series. The x-axis is time in seconds and the y-axis is amplitude of each click. The primary reason for the time series is to show the duration of the click, click rate, and interclick interval. Spectrum. The x-axis is frequency in kilohertz, and the y-axis is the amplitude, and it shows the energy within the call. I can see the frequency content in the spectrogram and spectrum of the call. The call starts a little above 40 kilohertz and goes up to about 95 kilohertz. We can determine the ICI of the call by counting the amount of clicks the beaked whale makes within 1 second. We determine from the time series that this whale makes 10 clicks per second. That's 100 milliseconds between each click. This is enough information to classify this as a Steineger's beaked whale. Individual click. The first graph is a spectrogram zoomed in on 1 click, and you can see the upsweep within the click. Both graphs are showing a fraction of a second, and it shows just 1 echolocation click. The time series confirms that it is a beaked whale because beaked whales have many cycles in their clicks. With all this information, we would reference our identification guide to confirm the species of the whale based on the click. Our identification guide is made up from information gathered from previously published data. This is a 24-hour plot of Steineger's beaked whale at the site KOA. There doesn't appear to be a diel pattern to when they click. Something of note is that there is no Steineger's calls the whole month of August. There are some big gaps in time between each click presence. Steineger's beaked whales aren't calling many hours of the week. Steinegers were present with an exception of a week in June and the whole month of August. Peak calling time was a week in May, in which they made an hour of calls, and a week in July, where they made about half an hour of calls. And this is at Site CB. It appears that Steineger's beaked whales at Site CB were more active during the spring and summer months. There isn't a particular pattern of presence for the 24-hour period. It appears that Steinegers have more hours of calls per week at the CB site. The peaks in calling hours never exceed 1 hour. There are some definite lows. The peak weeks are 1 hour at the end of May, 2 times in June, in July, and a week in August. There's a 3-week gap of no calls from July to August. We compared our analysis with the analysis of others' researchers' data. One thing of interest is that there seems to be a consistent absence of Steineger's beaked whale during the month of August for the years 2013, 2014, 2015, 2017, and 2019. The first report came from Debich et al. 2014. The second plot is from Rice et al. 2015. And the third plot is from Rice et al. 2018.
Please check to make sure that your microphones are muted if you're not speaking. Thank you.
In this day chart, Steininger's beaked whales and killer whales occur at the same time. Killer whale occurrence spikes from April to July. Steininger's occurrence stays the same throughout the year. Steininger's beaked whales at Site CB coexist frequently with killer whales.
Reese, will you read this again, please?
Yeah. There are 2 species of Baird's beaked whales, Berardius bairdi and the recently described Berardius minimus, which is darker and smaller than the more common Baird's beaked whale. The light blue areas on the lower right map show their range in the North Pacific Ocean. Baird's beaked whales produce clicks that look like other beaked whales, but they also produce dolphin-like clicks and some whistles. The whistles are pretty high frequency and difficult to hear over Zoom. You can listen to them on the Voices in the Sea website. We still aren't sure if it is possible to distinguish echolocation clicks from the 2 Baird's beaked whale species, so it's possible that our detections include encounters from both Baird's beaked whale species. This is an LTSA showing some Baird's beaked whale clicks. Some characteristics we look for in these clicks are that the majority of the intensity of the clicks are below about 30 kilohertz. Another indication of Baird's clicks are the band of energy you see around 10 kilohertz, where the red circle is located. Zooming in on an individual click will help further identify Baird's beaked whales. Here, we're showing 1 second of Baird's beaked whale clicks in the spectrogram at the top and the time series at the bottom. With 5 clicks within 1 second, Baird's beaked whales have an interclick interval of 200 ms. If we zoom in even further and look at one individual click, the spectrogram in the top panel shows the characteristic upsweep of beaked whale clicks. The time series in the center panel shows that the click has a longer duration and many cycles. Finally, the spectrum at the bottom panel shows that the peak frequency of Baird's beaked whale clicks are from 10 to 30 kilohertz. These are the different characteristics that help us identify Baird's beaked whales in our data.
It's my turn. This is a 24-hour plot from Site KOA showing our detection of Baird's beaked whales. In this data, we saw consistent detection of Baird's beaked whales throughout the day and night during May through the end of July. However, there is a gap of completely no presence detected through the middle of June to mid-September. This graph shows weekly presence of Baird's beaked whales. The x-axis shows months and the y-axis represents cumulative hours. There were Baird's beaked whales present for up to 20 hours per week from the end of May to the beginning of July, but then no detections for a couple of months in the middle of the deployment. This is a 24-hour plot of Site CB, Baird's beaked whales. We can see from this graph that Baird's were also very active and consistent through the months of May through early July. Then after that, there was a little activity until mid-September. There also does not appear to be any difference in Baird's click behavior between daytime and nighttime. This graph shows the weekly presence of Baird's beaked whales at Site CB. We saw a really consistent presence of Baird's up to 43 hours a week in June, but moving into July to August, their presence was dramatically reduced. But why? Beaked whales are sensitive to anthropogenic noise, so this could be a possible reasoning behind their disappearance. They also could have moved away from the recording site to search for food or avoid predators.
This slide shows buried beaked whales and anthropogenic noise at Site KOA, further offshore than Site CB. The dark orange shows the ship noise. The lighter shade of orange shows the echo sounder, and the purple is buried beaked whale sounds. As you can see, the black rectangle is when most of the detections of buried beaked whales occurred. In this range, detections of buried beaked whales only overlapped with ship noises 2 times. In conclusion, burieds were predominantly present during the beginning of the study at Site KOA, whereas further in the study or in the months, burieds barely make any sound, and that is where most of the anthropogenic noises are most common. Here are the results for the weekly presence of anthropogenic noise and buried beaked whale at Site KOA. The x-axis shows the months of the duration. The y-axis is calling hours per week. And as you can see, the red is the anthropogenic noise and the gray is the buried beaked whale. There were a few weeks where there's overlap between buried beaked whales and anthropogenic noise. In the month of August, there is a peak of anthropogenic noise in 1 week, and there were no beaked whales during that time. This 24-hour plot shows buried beaked whales and anthropogenic noise at Site CB. The x-axis is the hour of the day, and the y-axis is the day of the month. The dark orange shows ship noise, the lighter shade of orange is echo sounder, the purple is buried beaked whale sounds, and the teal color is mid-frequency active sonar. As you can see, the black rectangle is where most of the burieds activity was. Burieds overlap with anthropogenic noises multiple times here. They are even present during the month of June, where there is the most anthropogenic activity. And while we ran into one mid-frequency active sonar one time, and believe me, it wasn't very pleasant to the buried's ears. This is the weekly presence of anthropogenic noise and buried beaked whale at Site CB. The x-axis shows the months of the recording, and the y-axis is the calling hours per week. The red is the anthropogenic noise, and the gray is the buried beaked whale. As you can see, during the peak of the anthropogenic activity in June, there is a still significant amount of beaked whales activity. That activity drops off in July through September when there is also less ship noise.
Here we have killer whales and buried-beaked whales at Site KOA. Killer whales are shown in orange and buried-beaked whales are shown in blue. There are no times in the deployment where they can be heard at the same time. There are quite a few detections of buried-beaked whales in the beginning of the deployment, but as killer whales seem to be heard more often, buried-beaked whales activity decreases. Here Here we can see that there are multiple times early in deployment that we can see Baird's-beaked whales and killer whales present at the same time. It would be interesting to research what killer whale ecotype is found there and what that means for interactions of these two species. Here we can see that killer whales peak just before June, while Baird's beaked whale peaked late June. At the end of July, there's a decrease in both species. On the top, we have an older graph from 2013-2014 at Site CB. While comparing these data sets, we can see that in 2019, we detected Baird's beaked whales from May to September, when in the previous year, there were no detections during that time.
Now it's time to talk about Rizzo's dolphins, also known as Grandpa's greases. They are best recognized by their stocky bodies with blunt heads and no discernible beak. Due to fishnets, squids, and other dolphins, Rizzos are known for their scratches and scars. Males and females are usually around the same size, and their estimated lifespan is 35 years. Looking at where it says distribution of Rizzos, the gray represents where Rizzos can be found. They reside in almost all temperate and tropical waters around the world, from the Gulf of Alaska to the southern hemisphere of New Zealand. When we talk about their behavior in the water, Rizzos are usually found in groups of 10 to 30 dolphins, but some can be solitary or in loose aggregations. When foraging, their diet consists of fish, squid, prawns, shrimp, and cuttlefish. Rizzo's dolphins can deep dive to at least 1,000 feet and hold their breath for 30 minutes, but they usually make the shorter dives of just 5 to 7 minutes. The acoustic behavior of the species includes whistles, grunts, chirps, echolocation clicks, known as pulses, rapid series of clicks that have been described as barks, buzzes, and whistle burst pulses. From what I've just described, we know that they have a large range of different sounds.
Go ahead.
In that clip, you just heard Rizzo's echolocation clicks. However, their clicks contain energy above our hearing range, so what we had to do was slow them down to a quarter of their normal speed and to bring them to a frequency our ears can hear. When looking at an LTSA, which we pulled from our own data, we can find that there is a consistent and distinctive spectral peak and notch pattern evident in the clicks from Rizzo's. You can also see a horizontal banding is apparent. On an LTSA, the y-axis is frequency in kilohertz. The x-axis is time, and the very right side of the graph is showing the intensity. Looking over at the spectrum, the x-axis represents frequency in hertz, and the y-axis shows us the spectrum level. From this graph, we can see that the peak frequency for Rizzo's range from 22, 25, 30.5, and 38.8 hertz. This is a 24-hour plot showing presence of Rizzo's at Site CB. From this graph, we can see that there were 4 different detections that occurred mainly during the nighttime hours. This is important to note because the time of the day they echolocate can tell us what they might be foraging on. This plot shows weekly presence of Rizzos at Site CB. Weekly detections of Rizzos were all under 1 hour in July to August and between August and September. Unfortunately, we have no results from KOA because we didn't have any detections of Rizzos at KOA. So, yeah. It is conclusion time. Throughout our collected data, we can confirm that 5 different odontencies were detected. Killer whales, sperm whales, Steinegers, and beaked whales, and Rizzo's dolphins were found. Though through all of this collected data, we learned that there were more odontency activity in the continental slope at Site CB. Killer whales show a potential peak in activity during the summer. Sperm whales maintain a consistent presence throughout the data. Beaked whales showed short-term fluctuation in habitat use. And lastly, we found that Rizzo's dolphins showed little to no activity, but were most active during the night when detected.
Reese, you there?
I was muted and I didn't even realize it. A lot of people helped make this project possible. We'd like to thank the technicians at Scripps who built the harps, went out to sea to deploy and recover the instruments, and then process the data back to the lab so that it's ready for us to analyze. The funding to support this research comes from the U.S. Navy, Professor Bob Rex, and Mount Edgecombe High School. Thank you for your time and attendance. Now we will take any questions that you might have.
Yay.
I have a question.
All right.
Well, first of all, Jesse, when my dog was laying at my feet as you were giving your talk. In one of your slides, you had a whale whistle, and it was so high pitched, and that dog got up immediately when she heard the pitch. But my other question, I don't know if it'll be for John, Michael, whoever, and of course it's going to be COVID related. So next year, 2021, when you guys are actually down at Scripps and presenting, do you think that you'll see, things might look a little different between, say, March and July or August, and you'll say, well, nobody was sailing, ships weren't moving, and things might look a little bit different? Will you add any of that into next year's? Do you think you'll feel that would be a thing? John?
It'll definitely be interesting for sure to do that. I mean, not a lot of boats are coming around, especially during this time, so it'll be cool to see the big difference in the impact that COVID-19 had on marine life. Positive.
John, do you feel that as well? Do you think that there will be?
Well, we're asking that question kind of all around the planet, and up here, maybe fewer ships because there's less commerce. The Gulf of Alaska or Gulf of Mexico, maybe there are fewer air guns, and so the data we're collecting right now are really important for answering that question. So, but maybe I don't know this time next year, but within the next couple of years we might have an answer for that. But I think it is an important question. Yeah. So I had a question about on some of the plots, you would show presence of ships and presence of echo sounders, and there were times where there are echo sounders with no ship, and I would surmise that there's a ship. There's not an echo sounder out by itself, but what created that? Where you see there's an echo sounder, but there's no ship noise.
So that was
that was.
Go ahead, Jesse.
When there was a presence of an echo sounder, there was a ship also. When it said just ship, it was just the only thing that could be heard was like habitation noise, engine noise. There was no. So an echo sounder was a ship and an echo sounder.
I got it. I'm sorry, I misinterpreted that. Yes.
Oh no, it's all right.
Well,
that makes sense.
There was also a decision made in class that we would do manual picks for the echo sounders. And then we used the automated detector for the ship sounds. So some of those ship sounds may not have appeared through the automated detector, but we were able to pick out faint echo sounders.
Right. Yeah, and there's the one instance of the MFA sonar. I mean, I think it would be interesting, if there were more, you could start to look for patterns of what's present or not. But But it is nice that you have that one instance, and I think it's just a matter of collecting more statistics to be able to say, is there something there?
Mm-hmm.
Why so many sperm whales? That's like sperm whale heaven out there. I'm impressed. But anybody have an idea of why there's so many sperm whales?
We had.
Go ahead, Sophia.
We had an idea that since it was on the shelf, kind of where they were showing where the map of where Site CB and KOA were, that since there was that shelf there, it kind of gave them the advantage of feeding. I think Mahoney's showing.
There we go.
Yeah, they have a little advantage to feeding right there, as opposed to more open ocean.
Can I add one question to just follow up on John's question? Why the sperm whales? First of all, there's a beautiful presentation and just remarkable work you guys have done this year. Super neat to see. It's interesting that the sperm whale presence looked like it was pretty much the entire time at the shallower site at CB. And then there's these two really distinct peaks in sperm whale presence in, it looked like May and August out there in offshore at the KOA site. Any ideas about what might be going on there?
I'm not sure actually. Maybe Ann knows.
I would defer to our sperm whale and Gulf of Alaska expert, Natalie, actually.
That is a really good question, actually. I think it might be, what I've noticed in the Gulf of Alaska is that at least at a few of the offshore sites and the seamounts, it seems like the sperm whales increase towards the end of summer. So I think it might have to do with better foraging opportunities for them at the end of summer at some of the more offshore sites, while at the slope site, they seem to just be there all the time. And if it is related to foraging, I'm not sure why the beaked whales seem to be gone during those months when the sperm whales were there, because they could be foraging on similar things, and it seems like they leave in August, but sperm whales stay there and actually increase in activity. So that's something I'll hopefully look into further with my PhD.
And I'll just add, we have a few students who are really interested in how the oceanography is impacting the animal behavior. So if we have some returning interns next year, they can start to dig into what are the drivers of those patterns that we observed.
I wanted to second what Josh just said, is that the presentation was amazingly professional and thoughtful. So, I mean, you guys have done a great job. I mean, I know I've said this before, but it seems like every year the level of sophistication that comes out of Mount Edgecombe High School just, reaches a new height. So I'm really impressed. And it's a shame, in fact, that some of the people who have paid us to collect the data, they should have been on this call, and then we could say, okay, we're done. There's our report. Mount Edgecombe has done it for us. But it was incredibly well put together.
Well, thank you very much.
Yeah.
The coronavirus has really posed some new challenges for us and for all of you. But I'm just super proud of the students for pulling through and continuing to wake up early in the morning and work really hard. And I think the presentation turned out fabulous.
Okay, well, next year, got to do it again, right? No stopping now.
Let's do it in person next year.
That would be good.
Mr. Mahoney,